Hello, I'm talking to you. This is Goddess of Science Biology, and in our cellular structure and function series, this is part 2, microscope technology. So, microscope technology, we couldn't find cells, we didn't know about cells or anything about them until the microscope got invented. And kind of the mantra here is as microscopes got better, the knowledge of the cell increased. So Robert Hooke and Anton van Leeuwenhoek were kind of the pioneers in the cell theory, but they used a very, very simple microscope. It only had a single lens and it looked very strange. So here's their microscopes, kind of famous. You would put whatever you're looking at down here and hope that the light was okay, and then look through the eyepiece up here. So it was really kind of a glorified magnifying glass. Couldn't see most parts of the cells. Some things that they could see were cell walls, obviously, because Robert Hooke named cells because he saw the cell wall. Maybe you could see a nucleus, maybe a plasma membrane, who knows? But it had to be something pretty big for you to be able to see it with this simple microscope that Anton and Robert were using. The second type of microscope came along about the time that Schleiden and Schwann were doing their research on plants and animal cells. It was a compound microscope, which meant that it didn't have just a single lens. It actually had a pair of lenses or 3 lenses, and each lens magnified what the previous lens was seeing. So this is a very simple thing that's found in most high school science biology labs, and it's had a few improvements over the years, but basically it was kind of what Schleiden and Schwann were using doing their research. It had more than one lens, and what happens is this is where one lens sits, and then there's a second lens up here. So whatever gets magnified by this lens gets magnified again by the lens in the eyepiece. So this type of microscope magnifies things hundreds of times. This is the microscope that most of you use in science class. Most of the time, the highest magnification is about 400 times, which is pretty good. You see lots more parts of the cells. You can definitely see the nucleus. You can see the plasma membrane for sure. You can see these little green things in plant cells called chloroplasts. And this is a video on how the simple microscope works. If you want to look on our webpage, there'll be a link there for that video. So this is your standard microscope, and I'm going to kind of give you the overview of the different parts and what they do, and some do's and don'ts of the microscope. So nota bene, which means pay attention to this, always carry your microscope with 2 hands. When you're taking it out of the cabinet where it's kept, make sure you're carrying it with 2 hands. They're not light. They're not terribly heavy either, but just kind of make sure you got 2 hands on that puppy whenever you're taking it out. So this up here is the eyepiece of your microscope, and the lens in the eyepiece magnifies things 10 times whatever you're looking at. Please don't take the eyepieces out of the microscope if you can, because what happens is as soon as you take the eyepiece out, dust can fall in here, and then invariably it's going to settle in the lowest point, which is on the lens of the objective. So when you're trying to look at something, you're going to be looking at a whole bunch of dust magnified like 400 times. So don't take the eyepieces off. This thing here is called the tube, quite appropriately, because, well, it is a tube. The neck is down here underneath. This is what all the business end of the microscope is attached to. The nose piece is out here in front. And notice that there are these 3 things screwed into the nose piece. The nose piece can turn, allowing you to see different magnifications of using the same microscope. The low power objective magnifies things 4 times. So if you had the low power objective all by itself, you would be able to see that it's 4 times. But because it's connected to the rest of the microscope, it actually magnifies more than that. The medium power objective magnifies things 10 times, and the high power objective magnifies things 40 times. Now, these magnifications are just for the objectives you see here. Usually you can tell which objective is which by the length. The shortest one magnifies things the least, so that's the low power. The medium-sized one is the medium power, and the longest one is the high power. So if you want to figure out what the total magnification of something that you're looking at under the microscope is, you take the magnification of the eyepiece, and in our case it's always going to be 10, and you multiply it by the objective magnification. So if we were looking at something as we are supposed to be here with 40x on the objective and 10, that would mean that our total magnification would be 40 times 10, which would be 400 times. This thing here, this flat thing, is called the stage. Just like actors stand on the stage and perform, the microscope slide sits on there and kind of functions the same. Stage clips. Some microscopes have stage clips. Some microscopes have a mechanical stage. These are put here to keep the microscope slide from sliding off. You have the coarse adjustment here. This will move the stage the most. The thing about the coarse adjuster or the coarse focus is that if you use the coarse adjust with high power magnifications, the medium or the high power magnifications, you can actually move the stage so that the objective will actually break the glass slide, or the glass slide will break the objective lens. So you have to be really careful about using the coarse adjust. My rule of thumb is you only use the coarse adjust to get things into focus when you're using low power. After you get off low power and you're going to medium or high power, you never touch the coarse adjustment. So nota bene for the coarse adjust, always begin with the lowest power and the coarse adjust. That's going to move the stage the most, the quickest. So, and you need to do that when you're first starting with a microscope slide. Don't start on the highest power objective, because it will be like looking for a needle in a haystack. It's going to take you forever. You're going to get frustrated, and then you're not going to want to do this. Start with the lowest power. See if you can find what you're supposed to be looking at first on lowest power, and then sharpen it up using the coarse adjust. Never use the coarse adjust on medium or high power. You can damage the microscope, or you could damage the slide. The fine adjust, once you get it into focus with the coarse adjust, then you can just sharpen it up with the fine adjust. You're only going to use the fine adjust when you go to a higher power, medium or high power. You're only going to have to use the fine adjust to make sure that you see it. The thing, when you're looking at a microscope slide, try to get what you're interested in the very middle of the field of view. If it's off to one side and then you move to a higher power, it may not be in the field of view. But if it's right in the middle of your field of view, then you're definitely going to be where you're supposed to be. There's a little device underneath here. There are several different ways that these work. This is a diaphragm, and what it does is it adjusts how much light is coming through the hole. Sometimes it's easier to see things that are unstained if you have less light there blasting through. So the diaphragm, it controls how much light passes through from your light source down here up through the rest of the microscope. The light source is on the bottom here. All the microscopes that we have in class are electrical microscopes. You have to plug them in. Oftentimes people will tell me that the microscope isn't working. I'll look, there's no light source, and then I'll look to see if they have their microscope plugged in, and they don't. So before you come and ask for help with your microscope, make sure that it's plugged in. All of you have an electric light source on the bottom. You have no idea how lucky you are to have that light source. When I was in high school, we only had mirrors on the bottom, and then you had to like get the mirror position just right so it would reflect the light up into the microscope, and then you get everything focused. And usually by the time all of that got done, some idiot would come by and flick your mirror, and then you had to start all over again. It was really annoying. Be happy you have a light source. Technology is a fantastic thing. The last part of the microscope is the base. Please try to have the base flat on a desk or a countertop. Don't have it sitting half on something and half not. These are expensive, so make sure that you pay attention to where the base is sitting. Another type of microscope, which was invented in the 1940s, 1930s, 1940s, is the electron microscope, where a light microscope uses light to pass through the object to see it. Electron microscopes use electrons, those very tiny particles on the outside of atoms, and it shoots a beam of those through the object that you're looking at. So it uses a beam of electrons, and electrons are notoriously ill-behaved, so you have to focus them using magnets, and you can see incredibly tiny things. Magnifies things 500,000 times. Needless to say, we don't have an electron microscope at our high school. You can see the very tiniest parts of cells. When I worked at the Eye Research Institute in Boston, I often had to develop pictures that had been taken with an electron microscope, and it was just quite amazing. You can see things so tiny like ribosomes, the endoplasmic reticulum, centrioles. When I was in high school many, many years ago, they didn't even know about centrioles then because they hadn't discovered them yet. And it was only when electron microscopes got really, really good did they discover them. There are different kinds of electron microscopes. There are three that we're going to talk about. The first one is a transmission electron microscope, or a TEM, and that sends the beam of light directly through the thing you're trying to look at. So the section that you're looking at, the thing that you're looking at, has to be incredibly, incredibly thin. It has to be like 1/100th of the thickness of the hair on your head to be able to use an electron microscope to see anything. Here's a picture of a typical TEM picture. There's all kinds of endoplasmic reticulum here. There's a big structure, looks kind of like a mitochondria there. Got some Golgi going on over here. It doesn't really look like anything that we've ever seen before. So it's really, really quite amazing. Very easy to get lost. One thing I do want to tell you about the electron micrographs that you see, anytime you see any of them that's colored, that coloring is there because of the magic of Photoshop. It's not there because you can take color pictures using a TEM. The only thing you're seeing is the electrons, whether they get caught up or pass through certain parts of the subject that you're looking at. So TEM looks at the insides of cells, and you can really, really see the tiniest parts. A scanning electron microscope looks at the surfaces of things. So here's a nice picture of some common everyday thing that I'm sure you probably have seen once or twice. If you want to make a guess, now would be a good time. Okay, so what that is it's actually a piece of ash from like a cigarette or from a fire burning wood or something like that. There's also something called a scanning tunneling electron microscope, and this type of microscope looks at 3D images of really, really tiny things, almost as tiny as molecules. Like, for example, this picture here is a scanning tunneling electron micrograph of DNA image, and you can see the double helix kind of vaguely here. So it's very, very interesting. I have no experience with the scanning tunneling electron microscope for sure. Well, that's it for this time. Thanks for joining me. Have a great day.